Hello everyone and welcome back to Creation Myths. Today I am going to show you a part of a stream that I did a while back with Gutsit Gibbon and Dapper Dinosaur and Vishanti. We talked about a whole bunch of stuff, but the big thing was reviewing one of those Tor and Stadler videos. So the part I'm going to show you is about an hour long, and it's us talking about Dr. Rob Stadler talking about the Lenski long-term evolution experiment and getting basically every conceivable thing wrong. So I'm not going to spend more time on any intro here. You'll see how this works. We're going to go through clips of Stadler and Tor talking about the experiment, and we're all going to talk about what they're getting wrong and how egregious it is. And I, in particular, I was basically offended by just how sloppy all of this was. So here you go. Hope you enjoy. Thanks for being here.
Are you just ready to get into the Lenski stuff?
Sure, let's do it.
This is going to be so dumb. This is going to be so... Can I make some predictions first?
Please, while I'm finding it, predict away.
So first, it's not new because the gene in question for the Cit+ trait actually existed before. You're just duplicating it. Therefore, it's not actually a new trait, even though it is a new trait because trait equals phenotype. So it isn't. The aerobic citrate metabolism is a new trait. He'll say it's not. He's also going to say, and I have not watched this video, he's also going to say that it's actually devolution because they lost the ability to regulate that gene, which is wrong. The original copy, you could still turn it on and off. The new copy, you could turn it on and off, just different conditions. So you didn't lose that ability. That's just wrong. And then the third thing he's going to say is this is also turns out not even good because there was this one paper this one time that showed a higher mortality rate in the Cit+ population, and he might even use the phrase die off, and that is just a misunderstanding of how ecology works when your population density goes up. So that's what I think he's going to say.
Shall we?
Let's see how I did.
All right.
All is not lost here because they say, here's our champion. Our champion is the most complete investigation of evolutionary innovation comes from the long-term evolution experiment of E. coli. Some of you may be familiar with this. This is Richard.
Send out your champion!
Right away, that's wrong. That is not even the best. Like, that's wrong. And sure, these authors can say that that's like the best example of evolutionary novelty, whatever, but that's wrong because you've got one type of bacteria adapting to the same environmental conditions since 1988. You wouldn't expect a whole lot of novelty for that because they're adapting to the same conditions continuously. They're being pushed towards one fitness peak for one very particular set of conditions. So you wouldn't expect a whole bunch of new stuff to happen. You're going to get to that peak and then you're going to stay there. So like the whole premise that we're talking about here, that the LTEE is the be all and end all, like it's the final boss of experiments and evolutionary novelty, that's wrong.
Yeah.
That's just not right.
Jen, you're assuming that he knows what a fitness landscape is. I don't think he does.
Yell pause when you want to pause.
Lenski's work, essentially his whole career, it's like 30 plus years of work studying E. coli, about 80,000 generations that he has studied in the laboratory, all prospective, all repeatable, all directly measurable. You know, it's all high confidence stuff.
So how long is an E. coli generation?
They went through about 6 to 7 generations per day. And it actually could have been more, but they put it in a media that was limited, you know, and how much glucose was in there. So they kind of ran out of gas, you know, and they could have generated more than that.
Right. Yeah, because I think most bacteria will double every 20 minutes. And so they slowed it down to 6 or 7 a day.
Yeah.
So let's just ballpark that to 10.
These two, like, effing learn something first. You can, under super optimal conditions, if you get bacteria into the exponential phase of their growth curve, you can maybe get them below half an hour, maybe for a generation. But that is not most bacteria. That is the fastest replicating bacteria under optimal conditions, and only for a part of their growth cycle because they start off with a small population, very slow. They get to that exponential phase, but then it rapidly is going to peter out. So that rapid exponential phase is only going to last maybe a few hours. because typically you're doing it in a, in a limited media where they run out of food at some point. And even if you add more food, the waste products are going to start inhibiting their growth rate. So you can't just learn, creationists, learn literally anything about what you're talking about before you try to lecture the experts on it.
The biggest innovation that they came up with, and this is what the media gets all excited about, is after 33,000 generations, they saw the E. coli develop the ability to metabolize citrate. Citrate was another kind of, it wasn't even meant to be a nutrient, but it was part of the bath that they were placed in every day. And they found a way to use that as an energy source. And so they got faster growing and more productive.
Rob, this was the point. You're introducing things that they wouldn't eat, that they wouldn't have access to, that they weren't prepared to metabolize. And then they did. What do you think that tells us?
Don't worry.
He'll tell us what he thinks.
He's going to say that it doesn't count here in a second.
Yeah.
Of course.
That way. And so the paper we're reviewing, Stroud and Ratcliffe, they describe this as the evolution of citrate utilization was a major innovation in the context of the LTEE. So major innovation sounds like a patent to me. Let's see what they got here. So in Richard Dawkins.
So in the context of this long-term evolution experiment, right?
Yep, happening around 33,000 generations in.
Okay.
The only innovations can be like the ingenuity types that you could patent.
Yeah, that's why the patent framing is funny to me. Like it's gonna stay like this. It's gonna stay like this for the rest of the thing. By the way,
guys, basically, unless Terrence Howard could come up with it, it doesn't count as evolution. That's what I'm going. That's what I'm hearing.
So yes, that's why the comparison is so goofy to me.
Does Richard Lenski own the citrate metabolism patent now? Is that what this means?
I mean, to be fair, though, you actually can patent
dumb.
genomes in the United States, which is horrifying. So maybe he does. It's actually possible.
So he's going to gas up Richard Dawkins here, because of course he is, because that's the god of evolution now.
Of course. You mean, wait, hang on. Richard Dawkins, the Epstein friend?
Yeah.
Richard Dawkins?
From the Epstein files.
Noted transphobe and noted as Islamophobe, transphobe, and also potential sea salmon enjoyer.
Yeah, the guy that says he's an atheist and that, there's a God delusion, but then it is also somehow a cultural Christian and is fine with that. But it's mostly because he hates us.
I literally don't care about anything regarding Richard Dawkins except for the fact that he's in the Epstein files at this point. Like for me, now maybe call me controversial, but that's my litmus test. If you're in the Epstein files, I don't really it's great if you did good science back in the day, but I don't want to hear from you anymore ever again.
I'll give it a minor caveat in that if you're in there in an incriminating way, because remember, Rebecca Watson is in there, but not in a way that implicates her.
Because they're complaining about it, right?
Here's another thing. This is how we know time travel isn't real, because Stephen Hawking would have come back and removed himself from all of the files. Because he went to some parties. He's in there.
Yeah, he did.
Hawking, man. And I'm not even going to touch that. Okay, I'm going to let him keep going. He's going to gas up Richard Dawkins, and then we're going to the punchline here.
Dawkins described this as a beautiful demonstration of evolution in action, new information entering genomes without the intervention of a designer. So a lot of hype there. Mm-hmm. Richard Dawkins actually published this in his book, The Greatest Show on Earth, about 3 years before the actual DNA analysis came out. The DNA analysis of what happened.
Pause. I know something else. I have one more prediction to add. He's going to reference the Van Hofwegen paper, where Scott Minich, I don't know how to say it. The DI guy is the author. And they're going to be like, look how easy it is to actually evolve this if you force them to, right? If you impose a selective pressure as opposed to just let them do it. That's not the point, guys. But we'll get there.
I'm going to let them keep going here for a minute, because Jim's got to get something off his chest.
I suspect he didn't have anything else to highlight.
I think this was the biggest thing to talk about, yes.
Right.
So when they actually did the DNA analysis...
No, pause. No, that's wrong. Pause. What? No. From God, just read, learn, crack a textbook on literally freaking anything. These two clowns. That is cool. That trait The Citplat trait, which we'll go through, is cool, but that is not the single most important thing that came out of the Lenski experiment. The thing that, at least from a theoretical evolution perspective, that is like the big cool thing, is the relationship between mutation accumulation and population fitness and a bunch of other things, right? And so one of those lines, one of those 12 lines of bacteria in that experiment became what's called a mutator lineage. They evolved a mutator phenotype where
Super
they, now creationists would call that devolution, because what's happening is they're deactivating some error checking. So they accumulate more mutations per generation than the ancestral phenotype. That basically what they're doing is they're evolving a higher baseline mutation rate. And what happened in those mutator bacteria is their fitness compared to the ancestor, all of the 12 lineages got better. If you look at the graph, it like starts at 1 as the relative fitness, and it goes up to about between 1.7 and 2-ish as the relative fitness. But if you break it out by the 12 lineages, the highest one is the mutator lineage. It has the highest relative fitness of all of those lineages, showing how by exploring more sequence space, by trying out more different mutations, you actually find more beneficial genotypes than you would otherwise find. And over the term of the experiment, that is a net plus, which is the opposite of what creationists would claim because of, genetic entropy. And if you get more mutations, you're going to go extinct faster as you're going to break more things. And because, they would say those mutations are slightly deleterious, you can't get rid of them, they're going to accumulate, and eventually you're going to die. And just from a theoretical perspective, that finding has way broader implications and is way more interesting than the CIT+ finding, even though the biochemistry and the mechanism of the CIT+ stuff is really cool. But in terms of learning about how evolution works, the mutator lineage is way cooler than the CIT+ lineage.
Dr. Dan, are you telling me that there was a change in the allele frequencies of this E. coli population over time?
Over time.
But that would be evolution.
That would be the definition of evolution. Yes, shocking.
Remember that time you weren't sure if you knew more about evolution than these two? You already have demonstrated
you
that you do.
You did it.
There you go. Something that you're going to notice, and I'm sure I'll bring it up when I put this video together, I guess more near the beginning. But one thing that you're going to notice throughout the rest of this video and forever with all creationists until the heat death of the universe is that they don't know what fitness means, right? Dan alluded to this earlier, but this is going to be a reigning theme with the nature of these bacteria, because to them, any change in the genome is degradation of the genome, which means it's deleterious, which means it's a decrease in fitness. But that's not how fitness is defined. So they're going to get into this here in a second, and we're all going to have something to say about it. But please keep in mind that the definition of fitness is it's an individual's relative ability to reproduce and leave behind offspring compared to others within its population. And it's differential reproductive success. That's it. Anything that gives you an edge on that is going to necessarily make you a fitter individual.
That makes Sal sad.
Right. Yeah, Sal does not like this, but I don't really know what to tell the creationists on this. They want us to change the definition of fitness. It's been the same since Darwin. Truly, I can't help you. It's like flat earthers wanting to change the definition of gravity so that they can make the flat earth make sense,
Or level.
I love what there's a person named Wade in the chat who says, I bet that when younger creationists hear the word evolution, they think of something out of an Animorph cover.
Animorph, Pokemon, Digimon, whole 9 yards. Okay, I'm gonna let him keep going.
Off with this paper by Blunt et al., 2012. And in this paper, they tried to make a big deal of it. They talked about evolutionary innovation has 3 mutational stages. It's kind of dramatizing this situation. You have the first stage, which is potentiation. And that means you have some random mutations pop in that are kind of setting the stage, opening the door for innovation. The second stage, actualization, that's where the big mutation happened that started this new function, the big innovation. And then after that, you have the third stage, which is
Hey, pause real quick.
where some
He's so mad that it didn't sprout eyes, and he's like, they're just over dramatizing it.
This sounds like a model for the evolution of irreducibly complex traits, right there.
It almost does, doesn't it?
Put that out there, yeah, a little bit.
That's it.
Other mutations come in to optimize, to tweak it so it's just fully optimized. You get the full benefit of it.
Fully automated
Stroud and Ratcliffe
metabolism.
They cite that paper and they basically agree with this, and they say that the value of the long-term experimental framework here is unquestionable. By reviving ancient populations that happen to be frozen, but they revived them from the freezer at different time points in the LTEE before the evolution of citrate consumption, so before 33,000 generations, it was discovered that the ability to evolve this innovation was contingent upon several previous genetic changes. That would be your potentiation going on. And that occurred after approximately 20,000 generations. So there's a lot of discussion here about how complicated this was, how many mutations were needed, and how this innovation took some time and some complexity to it. But that's not the full story.
All right,
pause. You want to say anything before we get the full story?
Pause. So three things here. One, he keeps saying something very basic, very wrong, that the new trait was citrate metabolism, which is not the trait that evolved. The new trait is aerobic citrate metabolism. E. coli can metabolize citrate anaerobically, that is, in the absence of oxygen. The new trait is the ability to do so in the presence of oxygen. So that's one, and Rob has got this wrong multiple times, and he's saying it enough that we need to correct him on it. So that's one. Two, he's not disputing any of the stuff that was described there, which is good because that paper accurately describes what happened. Between 20,000 and 30,000 generations, there were two mutations that occurred. And without those two mutations, the subsequent gene duplication would actually be harmful to the bacteria. So you need these two mutations that are neutral in order for the duplication that allows you to utilize citrate aerobically to actually help you. If you don't have the earlier mutations and then the duplication occurs, it is strongly detrimental and rapidly selected out of the population. That is just an accurate description of what happened in this experiment. Okay.
So Dan, correct me if I'm wrong. Didn't he just say something to the tune of there's no telling how many mutations and had to happen and how complicated this was? So you're telling me that there were two? There were two, what they called in that paper, priming mutations or potentiating mutations, and then the gene duplication, and then you actually had subsequent mutations that occurred after that. That's in that, I forget the word, but the refined or whatever, the optimization that optimized that trait once the phenotype actually appeared. Boy, though, that doesn't sound like there's no telling. That sounds like there is telling. It sounds like
we know exactly what was required and the order in which it was required, in part because of the paper I predict he's about to mention that was able to evolve this trait much more rapidly. If I recall correctly, they didn't start with the original generation 0 bacteria. They started further along. They took a sample from further along and were able to go from there and get the CIT plus trait much faster by basically imposing a stronger selective pressure. So you had stronger selection operating.
But Dan, you don't seem to understand. First of all, nah. And second of all, God just gave the E. coli bacteria the ability to metabolize citrate. Duh.
There you go.
They were born,
but then that's and they're gonna, with that paper, he's gonna bring up the Van Hofwegen paper. If I'm gonna have to eat my hat if he doesn't do it, but I think he's gonna do it right here, where he's gonna say, look, this isn't actually that hard to evolve. This isn't a big innovation. You're able to do it really fast under different conditions. Yeah, okay. That doesn't mean you didn't need this mutation, then this mutation, then this duplication, then this mutation, right? That doesn't negate the number of things that needed to happen for it to happen. You're just saying that in a different population, under different conditions, it happened faster. That doesn't negate it. In fact, because this is an irreducibly complex trait, you're actually showing how easy it is to evolve irreducibly complex traits. So thank you.
All right, I'm going to let him keep going because I think we have about 5 minutes of this Lenski stuff and then 2 clips and then we'll be done. So I'm not going to make it under 2 hours. But
3 or 4 more hours, I think. 3 or 4.
Yeah.
We can do that in another hour or 2, yeah. Mm-hmm.
Yeah.
Because, first of all, we know that E. coli already had the ability to metabolize citrate in the presence of oxygen. In fact, almost every life form, any life form of any kind that has the citric acid cycle has the ability to metabolize citrate. So it's not a new functionality necessarily. The problem with E. coli was that if there was oxygen present, it wasn't able to pull it into the cell. It wasn't able to get the citrate into the cell so it could then process it. So Lenski's group, later they published here in 2014, they later discovered that this key innovation of being able to metabolize citrate when there is oxygen, it actually required only 2 duplications, 2 gene duplications. One is the
pause, equivocation. See, he just effed up because there are 2 duplications at play here, the CIT and the DCTA, right? Those are genes that got duplicated, and the duplicated copies have a different expression pattern than the ancestral copies. Those are required. But he said there are only 2 duplications to get this trait. You also need the point mutations that occurred earlier in the experimental history of that lineage, or the duplications are harmful to you. So you need the other mutations, but those are not duplications. Those are single nucleotide substitutions. It's a different type of mutation. I suspect, doesn't know the difference because he doesn't know what he's talking about. I think he just doesn't know the difference.
I think, interestingly enough, hearing you say that, Dan, I think he might have dug himself in a bit of a hole. We'll see what you think in a second.
Uh-oh.
D gene, and this is a membrane-based enzyme that can pull in citrate, but to do that you have to pay one succinate.
Are transport proteins enzymes? Is that a fair thing to say? Because that doesn't sound right to me, but what do I know?
Not really. It's not an enzyme. It's an antiporter. So it's a transmembrane protein. You could argue that if it is, I think this is active transport. So you could argue that it is an ATPase enzyme because it's probably using ATP. So just like you could argue that the sodium potassium pump is that you could say, well, it's not an enzyme, it's a transporter. Well, you could argue it's an ATPase, right? Because
but that's like almost ATPase. Everything in biology is some kind of ATPase.
Sure,
right,
yeah.
It's minor, but I'm sorry. Yeah.
Nobody calls it an enzyme, right?
Yeah.
So I kicked him forward a little bit just since he's reading stuff, but I'm gonna let him play again.
Okay. Mutations fully utilize citrate, indicating that earlier mutations thought to have potentiated the initial evolution of citrate plus the ability to metabolize citrate are not required for expression of the refined version of this trait. So things are looking simpler now. It's actually just these 2 genes duplicated and not this whole 3-stage process.
How is that simpler?
Also, yeah, that's more okay. So 2 gene duplications is more complicated than 2 substitutions and 1 gene duplication. Like he's wrong about this in every possible way, folks. He's getting everything wrong about this.
Can I? So I want to add it on this because this isn't my zone. That's why I brought these guys on.
Like it's my zone.
You do not need to be this well versed in a subject to discuss it. But you do if you're trying to debunk it, right? Like you need to know it inside and out if you're coming on here and trying to say that this isn't actually, as Rob Stadler has sort of painted it, just hype from the scientific community, trying to amp this up as if it's something that it is not.
I mean,
okay, let's hear from Jim.
Are we not all in this moment the reaction on James Tour's face?
Yeah, I've been doing that. I've been covering my mouth a lot to try to hide my spot. We got them both on a blank earlier.
I screenshotted it.
Good.
Knock the wind out of what Dawkins was so happy about?
I think so. Yes. But of course, those who strongly support the Tree of Life are going to defend this to the death. And a lot of ink has been spilled in this debate.
How would you know that though, Rob? How would you know the ink has been spilled if you haven't read it? It doesn't seem like you put eyes on the ink there. I just for transparency's sake, I'm not a biochemist, right? I made my happy little ass sit down and I read up on this all day today leading up to this exact conversation. This stuff that Rob is missing, some of it is on the Wikipedia. You know what I mean? All of it is in the paper.
Ouch.
So anyways, I'm gonna let him keep going.
So it's also interesting to note that the review paper that we're reviewing, this Stroud and Ratcliffe, they don't happen to mention this work of Quant. They just mentioned the previous work of Blunt from 2012. So they kind of missed this. And there's also even a later paper by Van Hofwegen et al., 2016, where they repeated this. And they made it super clear that it's only these two genes, need to be duplicated or amplified in order to get the job done. And their conclusion is kind of interesting. They say a more
So duplication and amplification are two distinct things. So you can duplicate a gene, you can amplify a gene. Those are two separate things. If you need both, those are distinct things that need to happen.
Different steps.
Right. Those are separate steps. Just want to clarify that for people. Those are distinct events. Like,
ready to keep going?
Go.
I am.
Accurate, although controversial interpretation of the LTEE is that E. coli's capacity to evolve is more limited than currently assumed. And remember, this is kind of the champion that Stroud and Ratcliffe has. This is the best they got.
Yeah.
This is the best they got. I don't remember reading that part, but that's fine.
Yeah, that doesn't ring a bell, but I didn't. Maybe I missed it.
Their DNA can...
I pulled up the Van Hofwegen paper, which Dr. Rob Stadler either didn't read or is deliberately misrepresenting, because I have this paper handy. I have highlighted it and annotated it because, of course, I have. And I just want to read a couple of short passages for everybody.
Do you want to share your screen, Dan?
I can just put it up on screen. Yeah, let me just put the paper up for y'all. And let's see if what the paper says is consistent with what Rob just said.
Before you do that, I want to point out Hawk SX, I'm not sure, says, since you guys mentioned Pokemon, why is James Tor acting like a Snorlax? That's a good question.
I was not trying to be like rude or mean or whatever, but he just looked wiped earlier. Like I've looked that way. I've tried to stream that way. It does not go well, but he's looking sleepy. He was looking a little sleepy. All right, I'm adding to the stage. Oh, I have to remove my media from the stage. Okay, hold on a second. I'm working on it. Okay, remove. And then
it's Erica's first time.
Yeah, it's my first time. Guys,
We're all
I'm new.
Professionals here.
I'm new to the job.
All right. So this is the paper, Van Hofwegen. Scott Minnick over here is a Discovery Institute guy. Just FYI, here I'm gonna make this bigger for everybody. There we go. That's good. So here's the line. Here's the line that I think Stadler is mostly quoting. Unsurprisingly, it is from the abstract, because of course it is. We said
always
amplified CIT-T and DCTA loci followed by DNA rearrangements consistent with promoter capture events for CIT-T. So Stadler doesn't know what that means, because those are separate things. The promoter capture event refers to the duplication of CIT-T. So it exists here in the genome, and then it duplicates, and now it exists where it used to and in a new place under the control of a new promoter. But the amplified CIT-T and DCTA loci, that means mutations happened first, and then the duplication happened, right? So what this is saying is consistent with the earlier paper that Stadler showed that he's saying this paper conflicts with. Now, you can get into more detail and say genetic analysis of Lenski's CIT plus strain showed amplification of the CIT-T locus, so mutations leading to higher expression. That's what that's showing, or higher activity. Could be either way, more of the protein or the protein works faster. Amplification coupled with promoter capture event, which is the gene duplication. Right? So now we've got mutations plus duplication, just like the 2012 paper said. And then a point mutation in DCTA at position -20 from the translational start is also a necessary mutation for efficient citrate utilization. It allows recapture of succinate export during citrate uptake. What that means is you also have to get the more succinate import to counteract the activity of the antiporter that pumps it out when you're importing the citrate. You need to bring that stuff back in because that's also a component of that biochemical system. So this paper, what we just read, is exactly consistent with the 2012 paper, and Rob either doesn't realize that, or is lying about it, or he didn't read this far.
This might be a good time for that poll that I suggested. Did Rob Stadler read the papers and fail to understand them? Did he read them, understand them, and lie? Or did he just not read them?
It's crazy to me too, because this is a paper that has DI guys on it. Like, you'd think that if it was your team, and there's so few papers from your team, that you'd at least be like, I mean, I'd make it my business to be familiar if there were that few of them, right? I mean, and he set out to talk in part about this specific experiment and the papers surrounding it. You gotta you gotta prep. I mean, it's not like he brought this up on the fly. It'd be understandable if this wasn't a presentation that he had prepared for ahead of time. But such is the nature of a Stadler tour.
Oh, I want to read one more line here, right here in between my two highlights, because what they're doing is they're picking, they say frozen fossils, so they're picking from an earlier point in the Lenski experiment and going from there. Right? Says amplification of CYT, so mutation that gives more CYT activity, followed by promoter capture, which is to say duplication. Now, creationists are always a little bit shady about this because the ones who know what went on don't explicitly say it, but they word it in such a way that implies that a promoter moved around rather than the gene itself. But what happened is the gene was duplicated.
That was the Carter. That was what's his name? Not right. James Carter. James Carter. That
was James Carter. Just totally biffed that one, even though he wanted to talk about it and then just face planted. Because again, all these guys get their stuff from creationist sources, so like they're not hearing good information. But then and then either of which allows aerobic expression. These mutations account in part for the novel CYT+ phenotype, and then you get into the other point mutation that's required. So again, like, it's right there in black and white. But I think the fact that Minnick is a DI guy is part of the problem here, because they don't say explicitly, like, there was a mutation that upregulated the gene, followed by the gene got duplicated, and now it's aerobically expressed. But that's a problem unless you can deal with the succinate, so you need to amplify that gene as well. That was the result of a point mutation, right? There's no paragraph that just says that. It's wordy and awkward, and that allows, I think, Stadler here to not get across accurately what is going on.
I think even then, even with the wordiness of this, it's hard. If you read this whole paper, I would imagine you would have a hard time coming away with his representation of it, right?
Yeah, here we go.
I just don't know that that's a doable deal. Oh no, they got the figure too. They got the figure that makes it really clear.
They got there it is. Like, there's your like right here. Like, here's your duplication right there, and then here's DCTA, and you've got mutations in that. So I could go through and read this. We're not going to be here all night while I parse every line of this figure legend. But the figure tells the story that you've got here's your duplication right there. CYT. There it is. Tandem duplication right there. CYT. And then where's DCTA? It's right there. What's going on with DCTA? Oh, you got a crossover event. So you're right. There's the duplication for DCTA. Yep. So you got the two duplications because there's your crossover, and then you also need the point mutations affecting each of the duplicated genes to make it work.
God, this
it's right there,
sucks.
guys.
It's like it's almost like an evolutionary biologist might be a little better primed to handle a paper on the long-term evolution experiment.
It helps to have some training in evolutionary bio to read this paper.
Shoot.
Yeah, but Dan, not too many people with that training would be willing to go on and lie to everyone like this. So that's the problem.
I don't know. Does DI have any biochemists on staff that might be better equipped to handle this kind of thing? Can we get Behe on the horn real fast? Maybe he can explain it to him.
feel
Well, no,
like
because then he'll be looking at a way that his pet baby, irreducible complexity, can evolve in front of his eyes. He doesn't want to do that.
well, shoot though.
Yeah,
like,
that's true.
James
Makes it
Tor
hard.
knows James Tor knows Cygart. Cygart's a biochemist. Get Cygart on. Or
Oh,
Cygart's got a
Get
biochem
Cy on
background.
here,
Like,
yeah, right.
have Cy come explain it to him. You know, I mean, Cy could break this down quite easily. Okay, Dan, am I good to boot
even-
you? Are you ready?
Switch the screen back. Yeah.
Even if
ready.
you got one of their own guys to explain this to them, they would still not admit that this confirms evolution, that evolution happened, that evolution is possible, that blah, 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 blah. It doesn't
No,
even
dude.
matter.
Dude, watch their long story short on this. I watched that too to prep for tonight. Like, they will say it. They will be like, it might seem like the fitness went up, but trust us, this isn't evolution. Like,
But
okay,
nah.
okay, but why? But why is it not evolution, right? And then it's redefining fitness, and it's misrepresenting the experiment, and it's not understanding the details. It's a lot of things. It's a layered cake of bad science communication.
And I don't even like cake.
Yeah, neither do I. All right, I'm gonna let him keep going for a minute.
They share DNA. Their DNA is probably a lot more prone to modifications than say a human DNA.
It's much more unstable, and of course, because they have such
Yes.
large numbers and reproduce
That's
so
not
rapidly,
true.
they can accomplish.
Yeah, do we have a citation for that, buddy?
Pause.
I need to see some evidence of that.
Bacteria have a lower inherent mutation rate than mammals. They have more mutations because they have short
No.
generations and giant populations,
Okay,
but they have
I
a
wasn't-
lower inherent mutation rate.
Hold on. I was, so when he said that, I was thinking to myself when I listened to this earlier today, I was like, oh man, I do not know enough about, I mean, I'm no geneticist. I don't know enough about mutation rates to say, but I bet
I do.
someone,
Mhm.
I'm having
I
someone
do.
on who does.
And they're wrong.
Mhm.
And they're wrong.
That's
Shocker.
That's hysterical. Oh my God, it's true. Oh, that rest in peace. That sucks. Oh man. God, it can't be easy having us looking over your shoulder all the time. Just what are you talking about over there, Rob? It was like your evolution? Let's take a closer look.
Hey,
What are you saying?
you guys talk about Lenski? Cool. What are you saying?
Hey, that's
talk about
really
Lenski?
For the record, we would just talk with them too. Like I would be so happy to sit down with either of these guys and talk about evolution, right? I'd love that. Dan would love that. Dapper would love that. Vishanti would love that. All of us would be so happy to do that. And I know we're mean on this kind of thing, but we're nice in person.
Very nice in
Because-
person.
Yeah.
At the very of the internet, we're all very sweet.
Right. But still, this is just two mutations.
Yeah, and it's even just duplications, you know. So
Still me
from the
too.
standpoint
Just
of innovation, it's kind of like
duplications.
It's
Oh
just a
my God. It's
duplication.
just duplicate. Freaking.
That's why that's when you said that earlier, Dan. I was like, yeah, he just digs this hole shovelful after shovelful.
This reminds me of when I was talking to Rebecca, Bird of Life, with Jackson, and she asked for an example of macroevolution, and her response when we asked her what would count was going from single cell to multicellular. And so we gave her two different papers of observed experiments in which multicellularity was induced in two different lineages separately.
Mm-hmm.
And then she was just like, nah, I
Nah.
don't think that counts.
No,
And it's like, why?
No, it doesn't count.
Not really a good reason. No real reason coming forward.
It's not
it just
checking
doesn't
the-
count.
it's not
Because
passing the
reasons.
vibe check. So
Yeah.
unfortunately, I'm gonna have to reject your paper. That's my peer review. Is I see how it feels.
Well, given
Let's
that
try it
younger
out. I feel
creationists literally have something called the cognitum, which is literally just, well, these two organisms don't pass the vibe check, so I guess they're not related to each other.
Yeah, that one drives me
Okay.
crazy. Like
Okay.
that works when you're careless Linnaeus, and not even him. That works when you're pre-Linnaeus, but that does not work. That's great chain of being. This is giving vegetable. Actually, this is giving mineral.
Yeah.
That's
It's
why,
like
by the way,
a-
Answers in Genesis has Capernae and Bovinae, which are both subfamilies of Bovidae,
Yeah, I know.
separate kinds in
I
their
know.
list of kinds, which is
Awesome.
it's a take. Yeah, it's a take.
No, I
Yeah.
know.
In the
Okay, I'm
chat,
gonna let him keep-
I think it's Dim Lep is saying, hang on while I move my goalposts.
Yes. I
mean,
need to
that's-
redo my short of the Kerbal Space Program rocket with a football
Please.
goal
We're just
post.
launching it into space.
Yes.
Yeah.
I need to
Good.
redo that one.
Good.
It will never lose its utility. Okay, I'm letting him go.
Like taking a page
By the way,
from a specific.
Dan?
Come on, Dan.
I just wanted to, I'm sorry, I had to look it up to make sure that I had it right, and I got a paper from Lynch. That's Michael Lynch, evolutionary biologist, looking at mutation rates. It's from 2006, is the figure that I'm familiar with that has the prokaryotes with the lower per mutation rate compared to the eukaryote. I just want to make sure that I cite my source on that. There are other sources that show them approximately equal, but the figure from Michael Lynch, anyway, just wanted to make sure that was out there.
Even if they were approximately equal, he did not say they're approximately equal. He said that
he said they're more unstable,
which is unstable. BS. Yeah.
In the chat, Fibonacci 8 writes, moving goalposts introduces its own heat problem.
Yeah, all the friction from all those goalposts, they keep moving.
Yeah. And it's still
the real cause of global warming.
And it still doesn't come close to accelerating the nuclear decay is the crazy thing. Nothing comes close.
Doesn't.
Okay, he's going. I'm letting him go. Nope.
Application document of a product, and putting it in the Xerox machine, you get another copy and you shove it in the specification. That's not very innovative. And in fact, just to take that a little further, that gene duplications like these ones we're talking about, they're known to occur very frequently. It's about 1,000 times more likely than a point mutation. When people talk about mutations, random mutations, I think a lot of people's brains naturally picture like a single nucleotide of DNA being switched. That's the mistake. But it's actually 1,000 times more likely that an entire gene is replicated, which is interesting.
Use the right words for things. Replication is when you copy your DNA when the cell is dividing. Duplication is when there's a recombination error and a region gets copied and there becomes 2 of that region in your genome. Use, like, creationists, you want to know why no one takes you seriously? It's because you don't even know what the right words are for things. Start there. Like, get the vocabulary that you learn in Bio 1 correct, and then we can start taking you seriously. But, like, start there.
Once again, young Earth creationists, stop acting like flat earthers, because you keep on doing it.
Like, use the right words. And I know creationists have been like, and Dr. Dan, he's so pedantic with the vocabulary. Words have specific technical meanings, and if you use the wrong words, no one knows what the hell you're trying to say.
Well, and it matters. It matters because of the goal here. If this was just 2 people discussing the long-term evolution experiment by Lenski, and they were just having a laugh, and they're interested in genetics, and they're interested in evolutionary biology, and so they're having a little conversation, that'd be fine. This is 2 people who are trying to show that the foundation of all of modern biology is BS, and that they have been hiding that from you, and that everything that seems to be support for it is a part of this concentrated hype machine to dupe the general public. You can't get basic stuff wrong, even if it's just vocab words, if that's your goal here. This, as Dan says, this stuff matters. It matters when you're trying to shift the paradigm.
Yep.
You gotta nut up or shut up on this.
It's one of those basic things where if you don't know what the position is, you can't refute it. Yeah. Period.
Yeah. Okay.
Actually, even more likely that a replicated gene gets deleted. So these life forms are quick to replicate a gene and then take it back, duplicate it, remove it. This happens very commonly.
And how many individual nucleotides are in a typical gene in this species?
I don't have the good statistics, but I'd put it in the range of, 1,000, 2,000.
I don't know, but let me wildly guess at it. Is what just happened there?
I mean, there's a gag just waiting to happen with. I mean, I don't know the statistics. Clip, chimp that bad boy and drop that whatever I needed. I don't know the statistics on that.
Yeah.
Yes.
It's some wonky post-flood stuff.
Some wonky post-citrate stuff, maybe.
Can I go back to my question about where the maned wolf fits in their forest of life?
No, he has no idea.
I mean,
he's not sure about speciation.
They usually use looks like as their main criteria, which is just vibes. But he also doesn't really think that you can use DNA to tell like nested hierarchies and descendancy and all this other stuff, which is a real problem for the legal system because there's a lot of people in jail based on like forensic evidence that are excited, I guess, that those are suddenly off the table. But then does he also think that paternity tests and DNA tests on the websites that are like, oh, you're 50% this and 16% that? Like, pick a lane, guy.
They don't want to talk about genetic similarity. And I'll tell you what, we didn't hear, I haven't heard a peep about the human chimp stuff from Stadler and from Tour since we bodied them on that last time. So I was glad to see that. But no, I mean, the answer is they don't know. They don't know. They don't have a model. Like, that's what's so rich about, to me at least, about Rob starting this by being like, okay, you know, we've got these two distinct models and they make different predictions. You don't have a model. First of all, this is an accommodation. You're hoping to take what we actively observe and retroactively have your model accommodate that. That's problem number one. But problem number two is that it cannot make sense of the data that we see, which is why Rob allows, like, for his model, anything, like straight down to last Thursdayism. Are Maine wolves related to dogs? Well, we don't really know. God could have done it that way, but maybe he didn't. I mean, who's really to say? Are you related to your grandma? I don't know. Maybe. God could have done it that way. Maybe not.
I mean, to be fair, I didn't watch my parents be born out of my grandmother. So yeah.
I mean, who's really to say if we can't see it happen? You know? Oh, wait, but we have historical records for that kind of thing, right? Wow.
I didn't see those get written.
Well, that's low confidence evidence, isn't it?
Yeah, in a court system, eyewitness certainly is, but not in Rob Stadler's six lines.
I didn't see any of these historical records actually being written, so I don't. Maybe someone made them up.
I'm gonna let him keep going. We're close to done with this Lenski stuff, and then I got 2 funny clips, maybe 3 funny clips, and then we're done.
It's a bit of a head scratcher why duplicating that entire gene of 1 or 2,000 is a common process. It kind of sounds like there's an intention there, because you're copying the whole gene and not just a piece, a fraction, or a single nucleotide. So
who's
here's
the
a quote from that
intender?
paper that,
you know,
obviously it's Jesus.
Oh, of course, silly me.
Yeah, Jesus is sitting there saying, "You know what this bacterium really needs? It's a new metabolic pathway. Let me just duplicate some genes real quick."
Yeah,
it was that or cure cancer. He decided to do the extra.
Let's do this one.
1943. What he just said was disproven in an experiment in 1943. It's the Luria-Delbruck fluctuation test, where they tested, this is so perfect because I literally just recorded a video on this a couple of days ago, where in this experiment, at the time, the question of how adaptation works was an open question. Over 80 years ago, in 1943, and they did the test. Is a certain trait the result of random mutation occurring as a population evolves and the members diverge from each other? Or is that adaptive trait a purposeful adaptive response to a new environmental condition? And this leads to a really clear prediction. If it's an adaptive response, you expect to see the frequency of that phenotype approximately equal across all your experimental populations. If it's due to random mutation, you're going to see big fluctuations in the frequency across your different populations. Some are going to have that mutation early, some are going to have it late, some are not going to have it at all. And what they found was the second thing. There were huge variations in the frequency of the adaptive phenotype in the bacterial populations. 100% conclusively showing that adaptive traits occur due to mutations that are probabilistic, not due to a purposeful, directed, adaptive, whatever word you want to use, response to a novel environmental condition. This was from 1943. And creationists, this is perfect, because in this video, I love this. I was like, this is continuous environmental tracking. But a lot of creationists say this without using those words. And here we go. He is describing continuous environmental tracking, but he will never in a million years use those words. But that's what he's describing.
Well, creationists are always on the cutting edge of science from 70 years ago.
I recorded that in the other order, and then I could clip that and show it.
He has his own word for this, right? He used this or his own words, I should say. He used them with me, and I think he alluded to it with Dan. He calls this designed mutability, right? He's talking about a tinkerer. He's talking about an active tinkerer who's actively, like PhD Tony said in the chat.
Yes.
Did they just suggest that every genetic mutation in every bacterium is a divinely ordained act of intervention? Kind of.
Yeah,
Sure
or at
seems
least
that
a
way.
lot of them. At least a lot of them.
Yeah, I mean that's to me designed mutability, and every mutation is an intervention by God. Like that, those sure do sound a lot the same
Also,
to me.
Dan, you're doing a lot of disparaging of continuous environmental tracking, but have you considered the fact that continuous environmental tracking has given us the amazing discovery that fish can get tans?
He hasn't considered that. Unfortunately, Dan.
Let me know
just
when
got
the eyes. Let me know when the eyes.
Yeah,
Yeah,
where are
I
the
don't
eyes?
care unless they
Where are the
sprout
eyes,
eyes.
Randy?
Where
All right.
are the eyes?
I'm letting him go.
Changes in gene copy number are among the most frequent mutational events in all genomes. And that's what happened in Lenski's E. coli. And there's a little more to the story too, because these E. coli that have quote unquote evolved to be able to metabolize citrate in the presence of oxygen are actually devolving. Because
here comes
they
devolution.
already have the ability to metabolize
Called it.
citrate, but only if there's no oxygen. So it's kind of like if you have a switch on your wall that turns a light on only when it's dark outside. So it's an automatic switch. That's kind of what the E. coli have right now, in that when there's no oxygen, the switch changes and now they can metabolize citrate. But now they have a second copy of this gene that can metabolize citrate when there is oxygen. So you have production of this gene with oxygen, production of this gene without oxygen. That's called constitutive expression, meaning that it's just constantly producing. And that's not always the best thing, you know.
So if he just says
that is wrong.
Evolution or so-called evolution, then that makes it not evolution. Checkmate.
So it's even worse than that though, because he's wrong. That gene is not under constitutive expression. Constitutive expression means it's expressed all the time. The promoter, where the gene landed to, is not a constitutive promoter. It's still regulated. You can still turn that gene on and off. It's just able to be on under a different set of conditions. But the promoter that I forget, it's like the RNK gene or whatever, is the gene that it is now it's kind of linked to in terms of expression. You can still regulate it. It's just regulated under a different set of conditions. I have tried to track down where creationists get this from, where they get this argument that it's now a constitutive gene, and I have not been able to figure out who said it first, but every creationist who talks about this makes that claim, and they are wrong.
Dan, get RJ down.
It's not. He's just wrong.
That's what he does.
This is also like what Dan is saying is true. Like, long story short, did this too. The DI did this too.
Yeah.
Like this, it's they're framing it exactly like this every single time. The DI, or I guess long story short, but they're affiliated with the DI. They even have like this little cheeky animation where the bacteria is producing the thing after being beaten over the head with a mallet. You know, I mean, it's and the idea is that this is negative. This could be like, this could theoretically, under some circumstances in some environments, be a bad thing. And they don't understand that that's fine. That's fine. That's okay. First of all, they're mischaracterizing it. But set that aside. That is how evolution works. Trade-offs, fitness landscapes. These are basic concepts that we teach to freshman undergraduates. You've got to get a handle on what fitness is, because otherwise you're shadow boxing. You're arguing with something that no one is saying. No one is saying that there's some kind of ultimate fitness, and he's gonna. Well, I'll do this.
Also, I gotta say this idea
of another quote from that's silly.
This idea of losing things isn't particularly useful, because virtually any change that ever occurs, you can contextualize in a way of losing whatever came before, right? Oh, you have lungs, you just lost the digestive system that didn't allow you to choke. Oh, you have arms and legs, you lost the ability to have fins, actually. Oh, you have an inner ear, I guess you don't have the ability to have those gill slits anymore, do you? Yeah, great, cool. Who cares?
Yeah, no, it's like that every time.
Yeah, and it's like, look, honest question here. If they want it to be rhetorical, then fine, but I'm genuinely curious. What does this do for their faith? Like, what actual function is this serving in bettering or deepening their faith?
It's a numbers game. It's just a numbers game, because we got to remember, this is a group of people for whom the highest expression of their faith is increasing the number of soul notches on their belt, right?
Right.
That is not something that is true for all groups of Christians, but it is certainly true for this group of Christians. These are the ones who are like, telling kids like, oh, how many of your schoolmates did you tell about Christianity today, Timmy? Oh, you didn't tell any of them, Timmy? Well, gotta be careful. I don't know. It doesn't sound like you have a saving faith to me.
Well, this can't be
that's this
evolution because then I don't get to store up treasures in heaven. So
I think it's more like anything that is perceived as potentially providing a naturalistic explanation for anything, right? Whether it's diversity or life in the first place, is a threat, right? If you're not with me, you're against me. And so from their perspective, that is something that cannot be true lest it poke even the finest little bit of holes in the type of faith that they have. Never mind that many, many, many Christians make it work, that these two things are perfectly copacetic with each other. But for these guys, it's all about denigrating the science of it all to sort of elevate and protect and reinforce the flock and reinforce that way of thinking. At least that's been my experience. And that
seems
was
like
my
for these guys, sounds like a skill issue.
By the way, Christopher Bearer
I just looked it up.
Oh, sorry.
I just looked it up. It is the RNK promoter. RNK is the promoter that the duplicated copy of CIT landed next to. And I just found 2 lists of known constitutive promoters in E. coli. RNK is on neither list. Thanks for playing. It's not constitutively expressed.
awesome.
Christopher Bearer of Christ is saying that science has hit a brick wall and it ain't going anywhere anytime soon. Tell you what, man, look, my email for my channel is public. Send me an email. We can set up a time to discuss whether or not evolution is going nowhere, whether science has hit a brick wall, whether rest in peace scientists. And I won't even bite, even though I do have a lot of
Sabina, is that your sock account?
No. Okay, we have 1 minute left in this Lenski stuff, then 2 funny clips, and we're done. Well, we have
Let's
super
do
chats,
it.
but
Let's do it.
1 minute left.
Lost control, basically, and production is kind of out of control. And Blunt, in a later paper, 2020, wrote this. However, we serendipitously discovered evidence of substantial cell death in cultures of a CIT-positive clone, such and such. These results indicate that the evolution of aerobic growth on citrate in the LTEE was associated with elevated mortality. So they're paying a price for having changed in this way.
Okay, this goes to 37:15, so there's still like 30 seconds to go. But Dan, are you 3 for 3? Did you get them all?
I just want to say,
Nice.
Are you not entertained? Are you not entertained?
Dan did not get now there's no way of knowing that he didn't pre-watch.
I didn't
But-
pre-watch. I didn't watch nothing.
If I'm just saying, if you choose to take him at his word, and I have no reason not to trust Dan, he's never lied to me before. He's got their number, man. That is
Bring
a
it, creationists.
knockout right
These
there.
guys
you're gonna
never
come
have
at
new
me.
ideas.
You're
Ever.
gonna come
They
at
do
me.
not have, they don't have
The
any
nerve, the
innovative
effing
claims.
nerve
They don't
to
have
come
any
at us.
novels. Explanations. It's all just the same crap repeated over and over and over, and they just try to mix it up a little bit.
The easy way to figure out why this is kind of a dumb argument is to think about this, right? I'm going to give you the name of 2 countries, and I want you to tell me which one has more deaths per day in that country, right? One of them is China, and the other one is the Vatican City. Which one has more deaths per day?
Yeah, China has more deaths per day.
Okay, so I guess it's way more dangerous in China than in the Vatican City because basically no one ever dies in the Vatican City. So you just
Almost
go there
never.
and you're basically immortal.
Almost never. You're immortal.
I just,
Yeah.
I gotta say,
We should finish it, and then we can.
Yeah.
So
We'll
many.
finish and then rip. I just want to, Dan, I want to give you your due, right? Like, he knew you were going to see this. That's all I'm going to say. He knew you were going to see this, and he still walked right into it. Homie
right
had the
into
map.
it.
Homie had the map and took a nosedive into the tar pits. But let's let him finish. Again, we're 37:15. That's where we're going.
So when we step back and look at all of this work that Lenski did, we ask ourselves, is this actually innovative? Well, Stroud and Ratcliffe described this as a major innovation. Richard Dawkins described it as, new information entering genomes. And what do you think? Would you say that this is something that deserves a patent? I don't think so. I don't think there's anything novel here. I don't think there's anything.
The goal isn't a
not
patent,
obvious
Rob.
that went on here. So
Right. It already had this ability under a slightly different condition, correct?
that's right. Not a patent.
No, no.
Okay, so this is just one of the 6 studies that I'm going to go through that came up.
So that's that's
right.
it for long-term evolution experiment from them.
Can I just give me a couple minutes here to just tear him a new orifice because
please.
So first, you cannot understand evolution without a reasonably basic understanding of ecology. You have to know some ecology for evolution to make sense. Those two fields go hand in hand. There's a concept in ecology called density-dependent mortality, where as a population becomes more dense, there are certain factors that cause mortality that increase in frequency. Things like disease transmission and waste accumulation, right? Those are larger problems in denser populations. Now, these E. coli, for people who aren't familiar, they're grown in teeny little flasks. They're about this big, right? And you've got like maybe 20 milliliters of liquid media in the flasks, and they're shaking in an incubator so the bacteria don't settle at the bottom. They stay suspended in the liquid. Now, the volume is constant. The flasks are not open. There's a beaker over top of it. So there's nothing going in, there's nothing coming out. It's the volume is constant. The citrate metabolizing populations were able to grow to a much greater population density because they had more food available to them than the other populations. Now, density independent mortality factors obviously are not going to change. They're going to stay constant because they are independent of the population density. But the density-dependent factors are going to become more severe due to the greater population density in that volume, in that culture of a constant volume. More cells, same volume, higher density, more mortality due to density-dependent factors.
Are you saying to me that if you have more things living, then more of them are going to die per unit time?
It's specifically if you have more things in a fixed area, you will have higher mortality per unit time due to density-dependent factors.
That seems very, very far-fetched, Dan. I don't know about that.
What can I tell you, Dapper?
I thought that the more things you got in a concentrated area, the longer they lived, which is why Mexico City is, of course, the place where no one dies.
So that's the first thing I wanted. I just wanted to explain, just what that observation is, and that it's straight down the middle with literally centuries of ecology, but maybe not literally centuries, but over a century of ecology. The second thing here is the fact that 2 hours ago or whenever I joined, I was able to say exactly what arguments he was going to make, down to referencing a specific paper he was going to cite and how he was going to misuse that paper. So I just, Rob, because I know you will, I don't know you're going to watch this, but I think you're probably going to watch this. I just want to say this, speaking directly to Rob. You come at us with this weak nonsense. This is what you're going to do. You're going to quote somebody saying this is the best example of evolution in the lab, that this explains how evolution works, and this is the best the evolutionists have got. And you are going to get everything wrong about this, from the nature of the mutations that led to the new trait, to the expression pattern of the duplicated copy of the gene, it's not constitutive, to the genomes are unstable. Oh, the die-off that occurs, the higher mortality, literally everything wrong about this. And it's not complicated stuff. You could literally Google constitutive E. coli promoters and see if RNK is on the list or not. And you couldn't even be bothered to do that. You just took what you've been told from other creationists who you trust, even though they're either as uninformed as you are, or they're lying to you, Rob. They're lying to you if they actually know this stuff. Because they are wrong about it, which has made you, sorry, I'm still ranting. I'm going to be done in a second, I promise. But it just, here's the thing. I don't presume to be able to lecture Dr. Rob Stadler, medical device inventor, engineer dude, on medical devices, because that's not my field. But here he is. He's going to come into my house and tell me we're wrong about everything. We have misrepresented these findings. We have blown them out of proportion. And then he has the nerve to do that while being wrong about every aspect of the thing he is presuming to lecture us evolutionary biologists about. How dare you? Get it together. That is just Reprehensible. Absolutely reprehensible, Rob.
I gotta point out too, all while having an untestable model himself.
Yep.
While ignoring the
good
8 months
Get
of
good.
conversation. Like, truly.
Yeah.
Wait, you're gonna spur a monumental crash out if you're not careful, Rob.
Saying that you don't have a model or that your model isn't testable is basically just removing yourself from the conversation as far as science goes, right?
Yeah.
Which, again, stop acting like a flurf who say, well, I don't have a model.
Yeah.
It's bold to do that
on the day of my daughter's wedding without a model.
I go off and the North Jersey comes out. Yeah, the North Jersey Italian comes out. Yeah, I know.
It's on the day of my daughter's wedding without a model to test.
Hey, take it easy, Tone. Take it easy, Tone.
Yay! I'm trying to move goalposts here. So, number one, get good. Number two, ironic, because all they do is accuse and project onto other people, but you just repeat what's been told to you. No one in science ever questions it. They just repeat what their teachers say or what they've been told to believe. Like, shut up. No, they don't. And three, all they have is, oh, you observed evolution? No, you didn't. Oh, you did? Well, that's not evolution. Repeat.
One of the things I think they don't really get is that virtually every single biology paper is to some degree testing some aspect of evolution. And you know how often evolution fails? It just doesn't. Just like every single observation of a new comet or asteroid is a test of how universal gravitation works. And you know how often universal gravitation fails? It doesn't. For the same reason, they're both accurate ways to describe the universe within the fields on which they touch.
Okay, wait, hold on. I misclicked. I'm sorry. This is also really nice, but I was trying to click on, where are you? There it is. Hey, I'm mischaracterizing
It's good.
Thank you
I'm lying in
here.
here, misrepresenting here.
All right. We got some funny stuff. We're gonna finish with some jokes. And then I'm coming back in.
It was the most stable population around.
I'm gonna come back in and thread through when I package this myself and touch on all of his other examples. So rest assured, Rob, if for whatever reason you end up catching this before my actual video on this comes out, I'm gonna go through the rest of your examples. So please wait with bated breath.
So that was Rob Stadler getting everything wrong about the Lenski long-term evolution experiment, and us on the panel having great fun talking about it. If you want to see that whole stream, that's linked down below, the original stream, and also the video that Erica made going into the parts of that Tor and Stadler video that we didn't have time to cover during the stream. That'll also be linked down below, so definitely check those out. Thank you all for watching. Before you go, remember to hit the various buttons, leave a comment, subscribe, all that good stuff, and as always, don't get fooled.